Hello, my name is Amparo López Rubio and I'm a researcher at the Institute of Agrochemistry and Food Technology from CSIC in Valencia, Spain, and I'm coordinating the Biocarb for Food project from the SUS-Food 2 call. So just to give you a bit of a background, seaweeds and seagrasses are extremely valuable and underexploited source of carbohydrates and bioactive compounds. However, nowadays at industrial scale, only purified carbohydrates are obtained, like agar or alginates, which are food texturizing ingredients, and the processes used to extract these carbohydrates are highly inefficient. Moreover, the remaining biomass, which is more than 50% of the initial material, is used as compost or simply disposed as organic waste. So in this context, the main objectives of the Biocarb for Food project, which we are carrying out in collaboration with Chagas, Nofima, the University of Hohenheim, and RISE, are first of all to explore alternative extraction techniques like ultrasound, microwaves, enzymes or their combination to make the processes more efficient and environmentally friendly. The second objective was to reduce the number of extraction steps to obtain less purified carbohydrates with additional functionalities, and finally to valorize the remaining biomass after extraction of carbohydrates, which are rich in lignocellulosic fractions, to develop biodegradable food packaging materials. So now my colleagues will tell you the main highlights of our project.
Hello, my name is Laura Gomez and I'm a PhD student based in Chagas, Ashton. I work for the Biocarb for Food project under Professor Brijes Tiwari's supervision alongside the technologist Eduarda Neves. Our main focus here in Chagas was to extract agar and alginates from seaweeds by using different novel extraction technologies such as ultrasound and microwave, and to compare them to conventional extraction methods. From our results, we could see that novel extraction technologies led to higher extraction yields compared to conventional extraction methods, and also that shorter extraction times were required in the case of novel extraction technologies. Therefore, we can conclude that novel extraction technologies have the potential to become greener methods to extract agar and alginates from seaweeds compared to conventional extraction methods.
One of Nofima's tasks in the Biocarb for Food project is characterization of two commercially important gel-forming seaweed polysaccharides. Why? So we can evaluate the performance of the novel polysaccharide extraction processes under investigation. Our focus is on agar and alginate. Specialized and dedicated quantitative methods are used for their analysis to yield information on their size, purity, process yield and composition. A prerequisite for successful analysis is access to advanced, complex and often very expensive lab equipment. This is not the type of analysis available at high throughput commercial analysis labs. These are the tools typically found in the research laboratory, such as at Nofima. For alginate, its compositional analysis is most often conducted using a nuclear magnetic resonance spectrometer, commonly known as NMR. A sample dissolved in deuterated water is simply placed into an NMR tube and the proton spectra collected in the instrument, often at 90 degrees. For these spectra, the major building blocks of alginate, manuronic acid and galluronic acid, can be determined, including information on the block lengths. For agar, the gold standard for purity, process, yield and composition analysis is known as reductive hydrolysis. In this method, a chemical known as morpholine borane is used in conjunction with trifluoroacetic acid. This mixture serves to break down the agar into its major monosaccharide building blocks of galactose and the special agar-specific monomer known as 3,6-anhydrogalactose. Under normal acid hydrolysis conditions, with acid only, 3,6-anhydrogalactose is destroyed. In the next step, these monosaccharides are reduced and then derivatized to make them volatile. They can then be analyzed and quantified by gas chromatography. If your lab is lucky enough to have a mass spectrometer attached to its GC, then one can also identify other monosaccharides such as methylated anhydrogalactose and galactose species. If you also have a flame ionization detector, then one can determine agar yield, purity and its composition. To measure polysaccharide size, we use a method known as multi-angle light scattering. Coupled to a liquid chromatography system, it allows us to accurately measure the size of alginate and agar. The main difference between the two analyses is that agar must be injected into the system hot to prevent it gelling.
In the Biocarb for Food project, we work with life cycle assessments to evaluate the sustainability of the products and processes that are developed in the project. And it's important to work with life cycle assessments already at this early stage of development to make sure that the products are sustainable and to find the environmental hotspots so that we can direct the development and optimization at the right places. And the first thing we did in the project was to do early stage life cycle assessments on the extraction processes for agar and alginate. And for both these products, we saw that the most important factor was the yield that could be obtained from the raw material. And of course, this is quite logical because the higher the yield is, the less inputs are needed to produce a certain amount of product. In the coming part of the project, we will continue to work with life cycle assessment, but to include the entire seaweed value chain, from the production of the raw material, and also include the valorization of the byproducts to biodegradable materials, which is one of the unique things of this project.